Everyone, I am Rajit Basak. Welcome to my channel Biology Buff. Today I am going to discuss about a very interesting topic, that is the enzymatic degradation of plastic. Before going to detail in this topic, we should know what's our plastic. Plastic are mostly synthetic polymer made by a chain of carbon atom with hydrogen, oxygen, nitrogen or sulfur, sometimes chlorine, which is present in the PVC built around the carbon chain. In this slide, we can see the structure of simple plastic, that is polyethylene. When ethylene monomer is get polymerized, it forms polyethylene. And these types of polyethylene mainly used to manufacture plastic bags, straw, etc. In 1862, Alexander Parker introduced world first man-made plastic, but fully synthetic plastic is pioneered in 1907 by Belgian chemist and marketer Leo Baekeland. Since 1950, plastic has been started to produce in a huge rate. Now, why plastic is so harmful to our world? As we know that the plastic is made up of a chemical which came from the production of planet warming fuel, so that we can say it contribute to the global warming. And we also know that the plastic is really difficult to decompose or recycled. Around 8.3 billion tons of plastic has been reported to be produced in last 70 years. So there are many harmful effects of the plastic. It basically can destroy the food chain of water bodies as well as land. The chemical composition of the plastic are so toxic and detrimental to our human body. It can cause immune problem, child development problem, cancer, etc. There are 7 types of plastic present in our environment. One of the most common type of plastic is polyethylene, which I have already discussed the uses and the structure of it. This plastic belongs to the class of thermoplastic. Now, what's a thermoplastic? Thermoplastic are the plastic polymer which is melted under heat after curing. Our next type of plastic is polyethylene terephthalate. This plastic also belongs to the class of thermoplastic. When ethylene and the terephthalic acid get polymerized through the formation of ester bond, it forms polyethylene terephthalate or PET. After this reaction, H2O is being released. These types of plastic mainly used to manufacture beverage bottles, etc. And this large aromatic ring is responsible for its toughness and strength. It is one of the main reasons for the plastic pollution caused by one-time use plastic products, which are then thrown away either on the land or on the oceans, as we can see in this picture. If the pollution of the plastic continue to grow this way, it can cause many harmful effects to our entire world. So, there are some microorganisms which can degrade plastic by their enzymatic reaction. As we can see in this slide, this microorganism firstly adhere the surface of the plastic and then secrete the enzyme which actually degrade the plastic. And after degrading this microorganism use the carbon source from it for their survival. Hence, they can flourish on the plastic waste when provided with optimal conditions necessary for their growth, like pH, temperature, etc. So, there are various types of methods which we have used to degrade plastic. Like, we can change the functional group due to the enzymatic degradation. We can also change the molecular weight of the plastic by enzymatic degradation. We can change the hydrophobicity. We can also change the surface topology. But most efficient method is to the decrease of the molecular weight by the enzymatic degradation. In this slide, we can see there are various types of microorganisms which secrete the enzyme which actually degrade plastic, like Ideonella sacchaensis secrete PETase, and PETase degrade PET type of plastic or polyethylene terephthalate. There are some fungi which secrete cutinase, lipase, etc. All these plastic degrading enzyme belongs to the class of hydrolysis. Now, before move on to the how these types of plastic degrading enzyme works, we have to know how does hydrolysis work. These hydrolysis enzymes are involving in a catalytic reaction which cause the breakdown of the chemical bond in the presence of H2O. H2O basically releases a H plus ion to form a nucleophile, and this nucleophile attack to break the bond. Before breaking the bond, it formed two types of intermediates, that is tetrahedral and trigonal bipolar, so that we can say this is the SN2 reaction. Now let's look into the how plastic degrading enzyme works. So basically the hydrolysis, these types of plastic degrading hydrolysis involve in a cleavage of the plastic polymer leads to the cleavage of the long carbon chain. This long carbon chain, and this reaction has two steps. All the plastic that are present in our environment are hydrophobic in nature. So the extracellular enzymes that are produced by various organisms firstly adhere to the surface of the plastic through the formation of hydrophobic interaction in the first step of the reaction and form plastic polymer and enzyme intermediate. Hydrophobic cleft is present in some hydrolysis near the active site of the enzyme can accommodate the hydrophobic group present in the polymer, thereby increasing the accessibility of the enzyme to the polymer. In the second stage of the reaction, active site of the enzyme participate which actually cleave the polymer and form some smaller monomer or dimer which actually accommodate by the bacteria or the microorganism and consume the carbon source for their survival. As we already come to know about how enzyme helps to degradation of plastic, now I am going to discuss further about a particular popular one, that is PETase. Now what is PETase? PETase is the polyethylene terephthalate hydrolysis or PET hydrolysis, which actually degrade the PET type of plastic and secreted by the Ideonella saccharensis. So this is the structure of PETase. Every hydrolysis have similar kinds of domain, but it contain two disulfide bridge, which you can see here, first one and second one, which stabilize and maintain the structural integrity of the protein, which is the key difference, because all other similar kinds of proteins have only one disulfide bridge, and this types of enzyme, especially PETase, belongs to the class of serine hydrolysis, means the serine residue present inside the active site of this enzyme. When the PETase containing bacteria are administered to the PET plastic, they secrete the PETase enzyme, which cause the PETase polymer bind to the active site of the PETase and allowing the reaction to occur. During the reaction, the enzyme break down the ester bond of the PET. At the end of the reaction, the enzyme PETase still completely exists, but the PET has been broken down into two subproducts, that is BHET and MHET. MHET is further broken down by MHETase and form TPA, that is terephthalic acid, and EG, that is ethylene glycerol. BHET, TPA and EG is the final product of the reaction, is then digested by the microorganism as their nutrition. So this pair of enzyme PETase and MHETase enable the bacteria to live on the plastic PET, that is polyethylene terephthalate, as the sole carbon source. Well, we don't have to worry about plastic waste anymore. What do you think? Well, we really do. PETase only degrade the PET type of plastic, so we can relax a bit about the PET plastic. Still, we have to avoid to use too many, because as we know that these types of microorganism will not able to cultivate everywhere where the PET exists. Also, PET is just one of the 7 types of plastic, and therefore we should still be concerned about other 6, in which some of them are excellently recycled, whereas some others are not at all recycled. And there also could potentially be many problems, such as inability to cultivate everywhere and their expensive cost. But PET hydrolysis is an extremely exciting molecule and has high potential for future sustainability and recycling. However, it is only the earlier days, it will still likely be a while before we used this microorganism to eat plastic. These are the reference from where I have read this topic. If you have any questions regarding this topic, you can drop comments. Thank you.